Welcome, dear listener, to a quiet corner of our world, a place where the veil between what we know and what we merely whisper about seems to thin. Tonight we embark on a gentle journey, not to distant lands or towering mountains, but into a realm often unseen, a microscopic universe teeming with secrets and wonders. Close your eyes softly, let your breath deepen, and allow the gentle currents of this story to carry you. We are going in search of a creature so tiny, so enigmatic, and so astonishingly resilient that it almost defies belief. It is known by many names, some affectionate, some hinting at its incredible nature: the water bear, the moss piglet, or more formally, the tardigrade. Imagine for a moment a being that has explored more of our planet than any human ever could. It has been found perched on the majestic peaks of mountains, where the air is thin and the winds are fierce. It has settled silently at the deepest, darkest abysses of the ocean, enduring pressures that would crush any other form of life. Its miniature body has nestled within the lush, humid embrace of tropical rainforests and clung steadfastly to the icy, desolate landscapes of Antarctica. Perhaps, even as you listen, a colony of these extraordinary creatures might be patiently residing in the moss growing on a forgotten rock in your garden, or perhaps on the damp, shaded side of a moldy garage. They are, quite simply, everywhere, a testament to life's boundless capacity for adaptation and endurance. But the ubiquity of tardigrades, as charming and widespread as it is merely the gentle introduction to their profound mystery. When you hear about tardigrades, there's one word that invariably accompanies their name, a word that captures the imagination and sparks a quiet awe: invincible. And in this reputation, there lies a startling, almost unbelievable truth. These aren't just robust creatures, they possess an intensity of survival that pushes the very boundaries of what we understand about life itself. Consider, if you will, the sheer extremes they can endure. Certain species of tardigrades can be frozen to temperatures barely above absolute zero, a frigid point where all molecular motion practically ceases. Yet they can emerge from this deep slumber seemingly unharmed. Conversely, they can be subjected to scorching heat exceeding 149 degrees Celsius, a temperature that would instantly sterilize and destroy most organic matter, and still, they persist. They can be completely dried out, losing almost all the water from their tiny bodies, shriveling into what might resemble a speck of dust, yet remain capable of reanimating years, even decades, later. And perhaps most astonishingly, these humble creatures have even survived the ultimate challenge: the vacuum and radiation of outer space. In these unfathomable conditions, the tardigrade does not merely survive in the active sense. Instead, it enters a remarkable state, a kind of suspended animation, where life appears to pause. It's as if they press a celestial standby button, awaiting more favorable times. And when those times arrive, sometimes after many long decades, they can be gently roused, coming back to life as if nothing extraordinary has transpired. This profound ability is so unlike anything we witness in our day-to-day lives, so far removed from our common understanding of biological limits, that it compels scientists to question the very definitions of what we consider alive versus what we deem dead. They challenge our perception of where the line is drawn, blurring it into a fascinating spectrum of existence. Beyond this perplexing resilience, even the basic anatomy of tardigrades presents its own array of puzzles, each piece adding to the quiet wonder of their being. In the vast, bustling, microscopic world, most creatures navigate their existence by wriggling and thrashing, by propelling themselves with rapidly beating cilia, or by lashing slender flagella. Legs, for creatures of such diminutive scale, are almost an unheard-of feature. Yet the tardigrade, this tiny water bear, walks, it ambles with 8 stubby legs. a unique characteristic that sets it apart in the unseen aquatic ballet of its peers. And the mystery of tardigrade anatomy only deepens when one ventures into the realm of their marine cousins. Here, in the hidden depths of the ocean, lie marine tardigrades adorned with structures so elaborate and bizarre, they are as dazzling to behold as they are baffling to comprehend. Some of these formations resemble miniature flagpoles extending from their bodies, waving gently in the currents. Others appear like inflatable, sticky feathers, catching the light in ways that seem almost fantastical. For many of these intricate and flamboyant structures, scientists still do not fully grasp their purpose. They simply exist, a silent question mark in the tapestry of life. For an organism so pervasive, found in nearly every corner of our planet, the tardigrade remains a profoundly mysterious being. We quietly ponder the reasons behind its evolution. Why did this microscopic creature, so small it needs a magnification to be seen, evolve to walk at all, when so many of its peers simply glide or wiggle? What is the function of those astonishing, almost whimsical morphological structures adorning the marine species? How is it that tardigrades have earned their reputation for invincibility? And what are the intricate mechanisms that allow for such extraordinary survival? And looking far beyond our terrestrial home, could their incredible abilities one day allow them to colonize the desolate expanse of outer space, or even to find a foothold in the harsh, alien environment of Mars? These are the questions that gently echo in the minds of scientists, inviting us to delve deeper into the quiet wisdom of these remarkable microscopic travelers. All tardigrades, in their essence, are aquatic animals. They require a delicate, shimmering film of water around their bodies, a vital sheath that facilitates their graceful locomotion and permits the subtle exchange of gases that sustains their life. However, it is a fascinating paradox that most of the tardigrades, known to science, are not found predominantly in deep bodies of water, but rather on land. They make their homes in the soft, verdant cushions of mosses and the delicate, often crusty patches of lichens that cling to rocks, trees, and soil across nearly all terrestrial environments. These land-dwelling tardigrades are affectionately known as limnoterrestrial species, and they are broadly classified within a group called eutardigrades. To truly appreciate these tiny marvels, one must understand their scale. They are incredibly small, typically measuring between 100 and 500 microns in length. To give you a sense of this, a human hair is roughly 50 to 100 microns thick, so they are barely visible, often appearing as little more than moving specks, even with keen eyesight. They are essentially only truly visible under the revealing lens of a microscope. The good news for anyone hoping to catch a glimpse of these elusive beings is that it doesn't have to be an incredibly powerful or sophisticated instrument. Even a relatively simple microscope can unlock the hidden world where they reside. Imagine, then, the quiet thrill of setting out to find one. If you recently found yourself in a new home, perhaps in a place like New England, where the environment is often wonderfully wet and adorned with lush, ancient mosses, the thought might gently arise, could I find a tardigrade here? A scientist, when asked about the best places to search, might quietly confirm that mosses and lichens are indeed prime hunting grounds. So, with a sense of calm anticipation, you might carefully gather a small clump of moss from a damp, shaded rock. Perhaps a gentle companion, a quiet dog named Betty, might even offer her silent presence, her soft tail wagging in quiet approval as you place the moss delicately into a petri dish. The hope is that, later, with a bit of patience and a simple technique of decanting, you might coax a tardigrade or two to emerge from their verdant hideaway. The story of tardigrades stretches back through an immense expanse of geological time, their origins appearing to trace all the way back to the Cambrian period. an astonishing era roughly 550 million years ago. To put this into perspective, imagine a world before land plants had fully emerged to carpet the continents in green, before the earliest ancestors of mammals took their first tentative steps, and long, long before the mighty dinosaurs roamed the earth. Basically, tardigrades have been around for an incredibly, almost unfathomably long time, witnessing epochs and transformations that have shaped our planet into what it is today. Despite their ancient lineage and widespread presence, scientists today still find themselves gently struggling to fit the tardigrade neatly into the grand, branching tapestry of the evolutionary tree. They occupy their very own unique phylum, a major taxonomic group, aptly named Tardigrade, and within the scientific community, there continues to be much quiet debate and thoughtful discussion about whether they are more closely related to the segmented, legged creatures we know as arthropods, such as insects and crustaceans, or to the simpler, unsegmented roundworms known as nematodes. This uncertainty highlights just how unique and singularly evolved these tiny beings truly are. Consider the work of a PhD student like Mark Mapallo, who considers himself a paleogridologist, a specialist with a particular, quiet interest in unearthing and studying ancient tardigrades. He explains that the oldest fossil tardigrades that scientists are truly confident about, those unequivocally identified as our tiny water bears, date back to the Cretaceous period. This places them roughly 90 million years ago, squarely within the reign of the dinosaurs. What's truly remarkable is how these fragile microscopic creatures have been preserved through such vast stretches of time. All of these ancient tardigrade fossils, he points out, are found encased in amber. Amber, for those who might not recall, is essentially fossilized tree resin. You're probably familiar with the sticky, often fragrant resin that oozes from certain trees when their bark is injured. A natural bandage for the tree. In the past, just as it happens today, unsuspecting insects, tiny plant fragments, or indeed tardigrades, would often become hopelessly trapped in this viscous golden substance, unable to escape its clinging embrace. On rare occasions, some of these ancient resinous blobs would fall into water, sinking gently to the bottom, where they became buried in the accumulating sediment. As these layers of earth settled deeper and deeper over millennia, the pressure and temperature began a slow, inexorable rise. These specific conditions caused the resin's organic compounds to undergo a process called polymerization, slowly transforming the soft, sticky resin into a hard, glassy, and remarkably durable material, the beautiful, transparent gemstone we know as amber. The organisms trapped within these amber prisons, whether they were insects, ancient plant life, or our tiny tardigrades, could be preserved with an extraordinary degree of fidelity, their intricate details held captive in a timeless embrace. Unfortunately, the delicate strands of DNA from these ancient tardigrades do not typically survive this immense stretch of time. DNA is a fragile molecule, and the very oldest traces of it we've ever managed to find date back only about 2 million years. So while we can't extract their genetic code, much of the tardigrade's external anatomy is beautifully preserved in the amber. And here lies another quiet marvel. Even though the oldest tardigrade fossil is a staggering 90 million years old, it still very much resembles a modern tardigrade. You could place it under a microscope today, and it would look almost identical to its living descendants. It's truly amazing, a testament to how fundamentally effective and enduring their basic external morphology has been, remaining virtually unchanged over such a colossal span of evolutionary time. This remarkable consistency is because the tardigrade body plan is, quite simply, extraordinarily effective. Let's gently explore this miniature architecture, starting at their head. Here they possess specialized mouth-like organs. These are not typical mouths as we might imagine. but rather equipped with piercing styles, tiny freaky little tongue-like structures. They use these styles with precision to pierce the tough cell walls of the plants, algae, or fungi that constitute their primary diet. Once a cell wall is breached, their esophagus, acting like a tiny powerful vacuum, gently sucks in the nutrient-rich cellular contents. These vital nutrients then quietly spread from their digestive tract to nourish the rest of their miniature body. However, not all tardigrades are peaceful vegetarians. Some species have embraced a more adventurous, predatory lifestyle, consuming entire living organisms. These include other microscopic creatures such as rotifers, nematodes, and surprisingly, even other tardigrades. These predatory tardigrades can actually have a significant, albeit unseen, impact on the delicate biodiversity of other microanimals in their environment. Scientists have observed that these hungry hunters can devour up to 56 nematodes a day under certain conditions. This voracious appetite can actually be quite beneficial for soil quality, particularly if those nematodes happen to be pests that parasitize and harm important plants. Moving slightly down the tardigrade's body, on its first segment, some species possess very basic eyes. These are not complex, image-forming eyes like our own, but rather simple structures made up of just a handful of visual cells. Their primary function is to detect light, helping the tardigrade orient itself, or perhaps sense changes in illumination. But as with so many aspects of tardigrade biology, there's a quiet enigma here. Recently, scientists identified multiple rhodopsins in tardigrades, which are light-sensitive proteins typically associated with vision. However, these opsins didn't seem to aid them in color vision, as they might in other animals. And even more strangely, they weren't particularly active in adult tardigrades at all. Instead, these mysterious opsins were most active when the tardigrades were still in their egg stage. What on earth, one might quietly wonder, would a tardigrade egg need visual opsins for? At present, no one has any idea, adding another layer of quiet wonder to their existence. Next, along the tardigrade's body, are three distinct trunk segments. Each of these segments proudly bears a pair of sturdy, stubby legs on either side, and at the end of each leg are delicate, yet firm, claws. The fifth and final segment of their body has another pair of legs, but these are uniquely oriented, facing distinctly backwards. It is thought that the unusual orientation of these posterior legs helps the tardigrade to grip onto surfaces with exceptional tenacity, almost like a miniature prehensile tail, offering a stable anchor in their often turbulent, watery world. The very existence of all these legs makes tardigrades truly strange and fascinating microscopic animals. Think about it for a moment. Creatures of the tardigrade's diminutive size rarely possess legs, and if they do, those appendages are typically not used for walking. For instance, water fleas, though they have legs, use them primarily for swimming and for sweeping tiny food particles into their mouths. Rotifers, creatures of a similar size, either gracefully swim through the water or inchworm their way along a substrate, pulling their bodies forward. And roundworms, those elongated, slender beings, simply wiggle and undulate their bodies to move. In this bustling microscopic domain, true walking is quite literally almost unheard of. One significant reason why walking might be so rare for such small organisms is the enormous challenge of overcoming viscous forces in water. For a tardigrade, moving through water is not like swimming for us. It's more akin to a human trying to walk through a vast, dense pool of honey. The water at their scale feels incredibly thick and resistant. Moreover, tardigrades must constantly contend with an extremely variable and complex environment. They might be trudging through syrupy films of water, clambering over tiny mountains of sediment grains, or navigating through dense, tangly clumps of plant matter. Yet, despite these formidable obstacles, Tardigrades utilize their 8 stubby legs to walk through all of this with an almost serene ease, the world over. Does the smallest legged animal on Earth possess some unique hidden method of employing its legs to overcome these persistent challenges? To unravel this quiet mystery, scientists turned to specialized cameras, meticulously observing the gait of tardigrades as they moved. What they discovered was truly fascinating. When tardigrades walk slowly, they exhibit a measured pace, lifting just 1 foot at a time, maintaining maximum contact with their substrate. As they pick up a bit of speed, their gait shifts. They lift 2 feet simultaneously, specifically those that are diagonal from each other across the body, always ensuring that at least 4 feet remain firmly on the ground, maintaining a steady balance. And even when they reach their fastest speeds, these tiny walkers keep a minimum of 3 feet on the ground at all times, not including their backward-facing legs. This careful, deliberate pattern ensures their stability, a crucial advantage in their unpredictable world. This walking pattern, which might appear somewhat random or uniquely peculiar to us at first glance, is actually not unique at all. The scientists realized, with a quiet sense of revelation, that despite the profound differences in their size and their fundamental skeletal structures, this particular way of walking was remarkably similar to that of much larger panarthropods, such as stick insects. Imagine, if you will, a creature about 500,000 times larger than a tardigrade and separated by a colossal 20 million years of evolution, yet employing essentially the same fundamental walking strategy. What, then, is the benefit of this specific type of walking for both stick insects and tardigrades? For stick insects, which navigate a world of precarious, pointy, jagged, and variable twigs and branches, always keeping 3 or 4 feet on the ground, provides an immense degree of stability. It prevents them from tumbling off their arboreal perches. For tardigrades, making their way through their own micro-world of variable complex terrain, perhaps climbing over a tiny pebble that feels like a boulder, or pushing through a tangled forest of algal filaments, keeping 3 or 4 feet firmly on the ground, similarly helps to provide crucial stability. So while tardigrades cannot run fast like a horse, or perhaps a micro-horse as one might playfully imagine, they are, most importantly, slow but incredibly steady. This striking similarity between the walking patterns of tardigrades and insects was a surprising and profound result. It is a discovery that may well point to the fundamental reason behind the very existence of tardigrade legs in the first place. It suggests a fascinating evolutionary possibility that arthropods and tardigrades might share a common ancestor, a creature from the distant past that already possessed legs wired to walk with this precise, stable pattern. Thus, this insight could be a significant piece in the ongoing puzzle of tardigrade taxonomy, gently nudging them closer to the arthropods on the evolutionary tree rather than to the nematodes. Further assisting tardigrades in their arduous journeys through thick and tangly microscopic environments are the specialized claws located at the end of their legs. Many tardigrades don't just have simple claws, they possess remarkably complex double claws on each leg. These intricate structures typically consist of 2 slender primary branches, which in turn give rise to 2 basal secondary branches. And within these secondary branches, there are usually 2 or 3 even finer hooks. These numerous finely tuned hooks are incredibly effective, allowing the tardigrade to grip onto its substrate with exceptional tenacity, ensuring they are not easily dislodged or carried away by errant currents or the jostling of their environment. The subtle differences in the number and specific shape of these claws are so distinctive that they serve as an important quiet key for scientists, allowing them to distinguish between the many different species of tardigrades. And there's something else profoundly fascinating about all the cells that compose these claws, legs, and body segments of a tardigrade. Every single cell that makes up every one of these intricate body parts is a cell that the tardigrade has possessed since its earliest developmental stages. Unlike most multicellular organisms, including ourselves, tardigrades do not grow by continuously dividing their cells to create more of them. Instead, their growth occurs through a unique process known as eutely. In eutely, the organism grows by the enlargement of its individual cells rather than by increasing the total number of cells through division. As a result, adult tardigrades of the same species will all roughly have the same characteristic number of cells, some possessing as many as 40,000. This remarkable phenomenon, growth by cellular enlargement, may offer a quiet evolutionary advantage. It's possible that this unique mode of growth decreases the risk of cancer or other problematic mutations that can often arise during the process of rapid and frequent cell division, offering them a subtle shield against certain cellular maladies. With all of this incredible intricate anatomy combined, it becomes clearer how tardigrades manage to thrive in virtually every corner of our planet. They inhabit the serene freshwater lakes, flow through the gentle currents of rivers, and reside in the quiet depths of ponds. They have established themselves on every single continent, from the bustling tropics to the desolate poles, a testament to their extraordinary adaptability and persistence. So if one were to acquire a microscope and venture out into the world with a sense of quiet anticipation, asking what are my chances of finding tardigrades, the answer, for many locations, would be resoundingly high. A scientist hearing you are in a place like Connecticut in New England might confidently declare, "Oh, there's a high chance of you finding tardigrades there. I feel like I always find them in New England." Imagine the quiet joy of such a discovery. You might diligently search through samples of moss, peering through the eyepiece, perhaps seeing little swimmy creatures that are not quite right, or cool, unusual forms that are certainly not tardigrades. And then suddenly, a subtle movement catches your eye. A tiny eight-legged form, undeniably moving with its characteristic, steady gait. Oh my God, you can totally see his little feetsies. Might be the excited, quiet whisper that escapes your lips. It's a moment of profound connection with a hidden world, a personal encounter with your very first tardigrade, a tiny emblem of life's boundless tenacity. But as challenging as it can be to find a terrestrial tardigrade, there is another type that is even more elusive, even harder to find, yet possesses an even more astonishing, almost fantastical appearance: the marine tardigrades. While terrestrial tardigrades are undoubtedly amazing in their resilience and unique biology, marine tardigrades are like something plucked directly from a Dr. Seuss hallucination, a testament to the ocean's boundless creativity. Marine tardigrades are usually classified as heterotardigrades, a group distinct from their land-dwelling eutardigrade cousins. They can be broadly divided into three major ecological groups, each occupying a specialized niche in the vast marine environment. First, there are the species that live on the slimy surfaces of algae or the hard, calcified plates of barnacles, sometimes functioning as ectoparasites, gently grazing on these living substrates. Then, there are the interstitial species, tiny denizens that make their home. Consider, for instance, a species named Tartis bubalis, which was discovered quietly residing in the Atlantic Ocean, near the remote Faroe Islands. This astonishing creature possesses remarkable posterior branches that extend from its rear end. These branches don't just extend, they branch and branch and branch again like a microscopic, intricate coral. And at the end of each of these delicate branches are unique, bubble-like formations that have the extraordinary ability to inflate or deflate. When they deflate, they become adhesive, allowing the creature to firmly anchor itself. But when they inflate, they can become buoyant, perhaps helping the tardigrade to gently lift itself into the water column. It is, as Dr. Bartels describes, like a Dr. Seuss-like character with these big balloons coming out the rear end. A truly crazy, captivating sight. And Tartis bubalis is hardly the only odd-looking heterotardigrade from the ocean depths. Some marine species are equipped with adhesive claws, allowing them to cling tightly to surfaces. Others possess delicate, wing-like structures, which are actually extensions of their cuticle, their tough outer covering. Still others have long, inflatable tails, adding to their whimsical appearance. Dr. Bartels expresses a particular interest in an interstitial species known as Bathyarchiniscus bulacodius. These are the tardigrades with tiny suction cup toes, enabling them to hold fast to the shifting, unstable sands of the intertidal zones. There are about 40 recognized species within this group, making it the most species-rich of all the marine tardigrades. One of the striking ways they vary from species to species is in their caudal structures, the formations at their rear. One species, discovered by one of his students near her home in the Outer Banks of North Carolina, has a tail that is truly remarkable. It stands like a tiny flagpole, almost vertically, from its rear end, and at the very tip of this flagpole-like tail, there is a large membranous bubble, like a miniature ethereal balloon. But what are these often ridiculous, often flamboyant structures actually for? For some of these formations, scientists have a decent, quiet idea. Some, for example, are described as thick, paddle-like structures. Others appear as more filamentous, hair-like appendages, and scientists know that at least on some of the paddle-shaped ones, there is a tiny pore at the end. This leads to the presumption, with pretty good evidence, that these are chemosensory, meaning they are involved in detecting chemical signals in the water, helping the tardigrade to smell its environment. The hair-like ones, conversely, are probably tactile, acting like delicate whiskers to feel their surroundings. However, for the truly crazy-looking caudal structures, like the flagpole tail of Bathyarchiniscus bulacodius, there is much less certainty, much more quiet speculation. One prevailing theory suggests that such structures might help them hold on tighter to their substrate, acting as a kind of anchor in turbulent waters. Yet, the flagpole, unlike the feathers of Tartis bubalis, isn't mucousy or sticky, so it might not actually adhere very well. Another intriguing possibility is that it provides them with some degree of buoyancy. Marine tardigrades, unlike almost all macroscopic marine animals, do not possess a larval planktonic stage, a free-swimming stage that allows for wide dispersal. So they don't have an obvious active way to spread across broad geographical ranges, and yet, in some cases, we know they do have pretty broad distributions. Perhaps they disperse passively, not by swimming. No tardigrades swim. They are all benthic, living on the bottom, but by being stirred up in the sediment and gently swept away by ocean currents. If such a structure could help them stay in the water column longer, even slightly, they would disperse further. My dream, Dr. Bartels muses quietly, is to figure out how to cut off the tails of the bulacodius with the flagpole, and then drop them and measure how fast they drop. Wouldn't that be a cool experiment? But experiments like this, as fascinating as they are, come with their own set of profound challenges. For as hard as terrestrial tardigrades are to find, that difficulty is magnified significantly when it comes to marine tardigrades. What about the marine ones are so much harder to study or harder to find? One might ask. Yeah, you got to have quite a bit of patience, Dr. Bartels explains. They can be really rare. They're much smaller in size than terrestrial ones, and you got to use a trick or two to isolate them from the sediment. One such trick is called the freshwater shock technique. This involves taking a bucket of marine sand, carefully collected from the intertidal zone, and then pouring a quantity of fresh water onto it. The sudden change in salinity triggers an osmotic shock in the tardigrades. This causes them to momentarily go into a state of physiological stress, loosening their tenacious grip on the sand grains. The shock stuns them, allowing researchers to then gently decant them through a series of sieves, separating them from the sediment. And if you're studying the deeper living marine tardigrades, the challenge intensifies even further. You would need to scuba dive into the ocean's depths, or employ specialized deep water collecting techniques, making such research incredibly demanding and accessible to only a dedicated few. For this reason, the vast majority of studies on marine tardigrades tend to focus on the interstitial species, those living in the more accessible sands. It's a truly specialized field. There's probably less than 10 of us in the world that are actively studying marine tardigrades, Dr. Bartels quietly states, underscoring the rarity of this expertise. And because so few people are looking, the potential for discovery is immense. Every place I've gone to do inventory-type work on marine tardigrades, we always find new species, he reveals. In a way, these new discoveries almost get in the way, as it takes so much dedicated work to describe and classify them, even when the primary interest might be an ecological question. It's a field rich with untold stories, with new forms of life constantly waiting to be unveiled. Today, scientists have already discovered and described more than 1,000 distinct species of tardigrades, and based on where we have, and, more importantly, where we have not yet looked for these tiny marvels, scientists quietly believe that there are many, many more still waiting to be found. A PhD student, like Mark Mapalo from the Philippines, might recount his own experience. He collected some mosses from the back of his building, cultured the tardigrades he found there, and was able to create monospecific cultures. And, as it turns out, the species he cultivated was in fact an entirely new species, a fresh discovery in the vast tapestry of life. So when asked, how many species do you think are still out there to be found? Is it in the tens or hundreds or thousands? The confident answer echoes, oh definitely. I would want to say at least 1,000. Thousands of new species out there. It's a humbling thought, that even in our seemingly well-mapped world, countless forms of life remain quietly uncatalogued, waiting for a patient eye to discover them. Tardigrades are often and quite dramatically referred to as invincible. This, of course, is not completely true, and it's important to understand the nuance behind this powerful word. In their natural environment, these tiny creatures are far from immune to the everyday challenges of life. They are quite likely to get eaten by larger microscopic predators, or injured by the jostling of their environment, or simply die of old age, just like any other living being. However, there is a profound and fascinating reason why they have earned this remarkable reputation. The story of their supposed invincibility truly began to unfold with groundbreaking experiments. In 1950, scientists made a stunning demonstration. Certain tardigrade species were shown to survive after being exposed to an astounding temperature of -273.3 degrees Celsius. This temperature is incredibly close to absolute zero, the theoretical point where all atomic motion ceases, a realm of ultimate cold. Later experiments continued to push the boundaries, revealing that these same species could briefly survive being heated to a scorching 150 degrees Celsius, a temperature that would boil water and instantly destroy most biological structures. They can also endure almost complete dehydration, losing nearly all the water from their bodies, shriveling into a seemingly lifeless state. The profound question that arises from these observations is, how can this squishy, unassuming little animal achieve any of this? And from an evolutionary perspective, what could possibly be the point? Why would a tiny aquatic animal, typically residing in a film of water, need to survive such extreme and seemingly irrelevant temperatures? The overarching biological method that allows tardigrades to live through such dramatic environmental changes is known as cryptobiosis, a truly remarkable state of suspended animation. When faced with unbearable conditions, the tardigrades enter what is called a toon state. Imagine, if you will, a miniature, shriveled nugget, a compact form that drastically reduces its surface area. To achieve this, they effectively shut their metabolism down almost completely, to the point where they are barely distinguishable from dead. Their life process is slow to an astonishing crawl, reaching as little as 0.01% of their normal metabolic rate. The fibers in their muscles lock into place, a bit like a sustained reversible rigor mortis, and often, a crucial part of this process involves losing an incredible amount of water from their body, sometimes up to 98% in some cases, becoming almost entirely desiccated. The specific trigger for this TUM state varies with the environmental challenge. When there isn't enough water, or when the temperature becomes too hot, a tardigrade will fully contract into this dehydrated TUM state. Freezing temperatures, however, trigger a slightly different specialized TUM state known as cryobiosis. In this cold-induced dormancy, tardigrades employ a sophisticated biochemical trick. They produce unique ice-nucleating proteins. These proteins work outside of their cells, effectively drawing water out of the cells before it can freeze and form damaging ice crystals within their delicate internal structures. This prevents the sharp shards of ice from rupturing their cells, a common cause of death for most organisms exposed to freezing. Also aiding in this miraculous transition are a unique group of proteins known as cytoplasmic abundant heat-soluble proteins, or CHEAS proteins for short. When the tardigrade experiences profound stress, such as severe loss of water, these remarkable proteins are produced in abundance. They then condense to form an intricate network of filaments, creating a gel-like substance that effectively encases and supports the cells. This gel provides essential structural integrity, acting like an internal scaffolding. Without the internal pressure of water inside the cell, which normally maintains its shape, the cells would simply collapse in on themselves. But this amazing gel stiffens, providing a stable structure that keeps the cells from collapsing. When conditions return to normal, and the life-giving water becomes available again, the CHES proteins are no longer produced, and the gel gracefully breaks down. The tardigrade then slowly takes in water once more, rehydrating its cells, and can gently wake up, resuming its miniature life, as if it had only paused for a moment. And what's even more impressive is the sheer duration for which they can survive in this state of suspended animation. They can endure this seemingly lifeless existence for decades. Researchers who collected samples of frozen moss from Antarctica in 1983, for instance, stored these samples at a constant -20 degrees Celsius for more than 30 years. Then, with a sense of quiet anticipation, the sample was carefully thawed. From this ancient moss, 2 tardigrades were gently extracted, affectionately named Sleeping Beauty 1 and Sleeping Beauty 2. They were provided with algae for food and closely monitored. Sadly, Sleeping Beauty 2 woke up but perished on day 20. However, Sleeping Beauty 1, after a full 9 days of recovery, finally awoke. It slowly began crawling around the petri dish, a tiny miracle stirring to life. It didn't eat until day 13, but by day 23 this ancient survivor was actually laying eggs, and continued to do so 4 more times, demonstrating a complete return to its biological functions. These results clearly show that the recovery process is not always easy, and that not all tardigrades survive. They are not, in fact, invincible in the absolute sense. It's also crucial to understand that tardigrades are not true extremophiles. Extremophiles are organisms, typically bacteria or archaea, that thrive and grow exclusively in extreme conditions. such as the superheated waters around hydrothermal vents in the deep ocean. Tardigrades, on the other hand, are considered extremo-tolerant. They can survive these extreme states by going dormant, but these are not the conditions in which they truly thrive, grow, and reproduce. They simply have an extraordinary ability to endure until more favorable, milder conditions return. And despite their widespread fame for extreme survival, tardigrades don't even hold the record for the creature that can survive the longest in a suspended state. Just in the last couple of years, some remarkable papers have emerged, almost making tardigrades look like mere novices in comparison. Right now, the only tardigrades that we know for sure, the length of time that they've survived in cryptobiosis, is a matter of like 30-40 years, maybe 50 in one study, one expert notes. Contrast this with two groundbreaking papers that came out over the last couple of years, focusing on rotifers and nematodes. These studies examined Siberian permafrost ice core samples, dated to an astonishing 26,000 years and 43,000 years ago respectively. They found both nematodes and rotifers preserved within these ancient ice cores. And when these unimaginably old samples were brought back to room temperature and gently immersed in water, these tiny creatures successfully came out of cryptobiosis, resuming their lives after tens of thousands of years. As far as we know, tardigrades don't even come close to those numbers. However, it's likely we simply haven't found the evidence yet. I believe it's only a matter of time before tardigrades are found in those samples as well, a scientist quietly asserts, because we know biochemically they have at least as many tricks as the nematodes and the rotifers have. But why, one might quietly ponder, did tardigrades evolve to survive such extremes at all? Surviving near absolute zero or intense radiation seems like a profound case of evolutionary overkill for a creature often found quietly munching on moss. It's likely because the tardigrade didn't evolve specifically for these dramatic headline-grabbing scenarios. Rather, these incredible abilities are a fortunate side benefit that stems from what the tardigrade did evolve for, a more mundane yet equally vital adaptation. Scientists now believe that tardigrades initially developed their extreme response to environmental changes in the ancient seas and oceans of a very, very old world. The ability to enter the tunt state may have first appeared not as a response to dry places, or super cold places, or super hot places, but as a crucial adaptation to fluctuations in salt levels in the sea. Imagine them living in the intertidal area, that dynamic zone where the tides ebb and flow. When the water surrounding these ancient tardigrades increased in salt concentration, perhaps due to evaporation or a shifting environment, the water inside the creature's permeable cells would have been pulled out through osmosis. Osmosis, as you might recall, is the passive movement of water from an area of lower solute concentration inside the cell to an area of higher solute concentration, the salty seawater. This natural process would have profoundly dehydrated the tardigrades. They would have needed some efficient way to manage the drastic dehydration caused by these shifts in salinity. The survival strategy they quietly arrived at was a form of cryptobiosis known as osmobiosis, a specialized tunt state for coping with osmotic stress. And this ability, honed over millions of years to survive overly salty conditions, serendipitously became an incredibly effective way to survive completely dry conditions as well. As tardigrades began to colonize new environments, venturing into terrestrial settings like sometimes temporary puddles and streams that would periodically dry out, this dehydrated nugget state became absolutely crucial for their survival. Furthermore, this capacity for desiccation and cellular protection also conferred a remarkable resistance to both extreme cold and extreme heat, allowing them to spread and flourish all over the world, establishing their microscopic presence on every continent. Often their inherent hardiness and resilience allow them to be the very first ones to venture into new, challenging environments. Scientists sometimes refer to tardigrades as pioneer species. This means they are frequently the initial forms of life to establish themselves in a harsh environment, a quiet vanguard of biodiversity. Whether it's the freshly cooled and hardened lava fields after a devastating volcanic eruption, or the toxic, degraded soils that result from intense coal mining, tardigrades often arrive first. They begin their miniature lives, quietly eating, mating, and becoming a food source for other microanimals. All this biological activity, small as it seems, gently sets the stage, paving the way for an ever-increasing number of creatures to enter and colonize that environment, slowly rebuilding the ecosystem from the ground up. These pioneering tendencies, combined with their incredible ability to survive almost anywhere, lead scientists to ponder the next grand question: can tardigrades survive space, or even one day colonize new planets? Space, hypothetically, represents the final, ultimate environment to colonize, a realm that almost no known organism can withstand, due to its punishing vacuum and intense unfiltered radiation. But tardigrades, as we've learned, are not most organisms. So in 2007, scientists, driven by a quiet, determined curiosity, did the natural thing. They gently sent them up there. The tardigrades hitched a ride on the European Space Agency's Photon M3 mission, an unmanned mission that carried 40 different experiments into low Earth orbit for a period of 12 days. Among these experiments were 3,000 tardigrades, representing 2 different species. They journeyed into space in their dehydrated chicken nugget ton state, already prepared for hardship. Once in orbit, they were exposed to the harsh vacuum of space, as well as to both UVA and UVB radiation from the sun. On Earth, our atmosphere, particularly the ozone layer, acts as a protective shield. UVC rays, the most dangerous, are almost completely absorbed. About 95% of UVB is also absorbed, but enough still reaches the surface to cause sunburns. UVA is the least dangerous, but still slightly increases the risk of skin cancer. For these specific experiments, researchers designed different exposure scenarios. Some tardigrades were exposed solely to the vacuum of space. Others were exposed to the vacuum of space combined with UVA and UVB radiation, and a third group was exposed to all types of radiation, including the more powerful UVC. The results were profoundly revealing. The tardigrades exposed only to the vacuum of space did remarkably well, demonstrating their inherent resilience to a complete lack of atmosphere. Those exposed to all types of radiation, unfortunately, all perished, indicating a limit to their invincibility. But significantly, some of those exposed to the vacuum along with UVA and UVB radiation did survive. The tardigrades that survived these combined conditions represented a monumental achievement. They were the first animals ever recorded to survive the combined exposure of vacuum, cosmic radiation, and UV radiation in the harsh environment of outer space. This incredible feat is largely attributed to a special set of genes they possess, aptly named damage suppressor, or DISP genes. These genes code for a unique protein that can form a protective bubble wrap around their precious DNA. Ongoing studies suggest that by binding directly to the DNA within the nucleus of their cells, the DISP protein acts as a physical shield, effectively preventing damaging radiation waves from directly hitting and fracturing the delicate DNA strands. It's the kind of thing that makes scientists quietly contemplate profound possibilities, wondering if these genes could one day be applied as a protective therapy to other animals, or even, remarkably, to us, helping to shield human cells from radiation damage. Scientists have even taken the bold step of inserting the DISP gene into human cells, using a virus as a carrier. They then fired X-rays, a form of radiation, at these human cells. They found that the DNA in the edited human cells was an impressive 40% less damaged than the DNA in cells without the inserted gene. While this is a long way from practical application, it offers a tantalizing glimpse into how these remarkable genes, inherited from a tiny water bear, might one day be used to protect astronauts venturing into the deep, radiation-filled expanses of space. Beyond protecting human space explorers, experiments on tardigrade survival in space might also gently open up entirely new avenues for space colonization. Mars, our mysterious red neighbor, has long been envisioned as a potential second home for humanity, but it is an unforgiving planet, incredibly cold, largely unprotected from intense radiation, and very, very dry. These are precisely the conditions that a tardigrade in its tunt state can manage with surprising ease. We already know that moss and lichen, which are also pioneer species much like tardigrades, can survive in very harsh environments here on Earth. Researchers are actively investigating whether genetically enhanced versions of moss could be engineered to grow in even harsher alien environments, and still provide good habitats and food sources for tardigrades. If such a scenario were to become feasible, these humble organisms, a starter pack of moss and tardigrades, could potentially be the first living pioneers, helping to bring life to new planets like Mars, initiating a gentle cascade of biological activity. This is all theoretical for now, a quiet dream for the distant future. There remains one vital element missing, an absolute necessity that even the indomitable tardigrades cannot live without in the long term: water. On Mars, the little water that does exist is largely trapped in ice beneath the surface, or bound within minerals in the planet's dusty soil. But this wasn't always the case. Billions of years ago, Mars used to be a very different place, covered in expansive oceans, a blue planet in its own right. And just a few days ago, the Perseverance rover made a truly significant discovery, a quiet whisper from the Martian past. The robot came across a viney rock on July 18th, a specimen that could profoundly change the course of planetary science forever. The rock bears clear evidence that water once ran through it, carving its intricate patterns. And even more excitingly, the rover detected carbon-based molecules within the rock, along with compelling evidence that life-sustaining chemical reactions may once have occurred there. And perhaps most excitingly of all, the rock is covered with what appears to be tiny, distinct leopard spots. On Earth, similar leopard spots in rocks are often a telltale sign of fossilized microbes, microscopic traces of ancient life. While we won't know for sure until this precious rock makes its long journey back to Earth, to be meticulously studied by scientists, the quiet possibility that evidence of Martian microbes might be found is agonizingly, exhilaratingly exciting. It leads us to wonder, in the calm of the night, what those microbes would have been like. Would they have been similar to the life we know on our own planet, perhaps akin to tardigrades, or nematodes, or bacteria as we understand them? Or would they represent something entirely new, a unique branch on the cosmic tree of life? When the story of this Martian discovery quietly broke, it was immediately clear that this was the kind of news that would quickly become sensationalized, amplified, and often distorted by various media outlets. It was almost a certainty that many news articles would get the facts straight up wrong, eager for a dramatic headline. In such moments, it becomes incredibly valuable to have tools that help us wade through the headlines, to find the best, most reliable sources, and to gently pull back the curtain on media bias and factuality. Imagine, then, a quiet, digital space, a website and app, designed precisely for this purpose, like Ground News. Here, every story comes with a visual breakdown, a calm and objective assessment of its political bias, its reliability, and its ownership, all meticulously backed by ratings from 3 independent news monitoring organizations. With such a tool, one could instantly see that yes, the headlines about the Mars discovery were indeed all over the place. Some headlines boldly claimed that NASA scientists had discovered actual life on Mars, while others were misleading at best, stretching the truth to capture attention. In contrast, the ground news headline summary of the story provided a much-needed dose of calm, factual clarity. No, NASA hasn't found life on Mars yet, but the latest discovery is intriguing. This gentle framing allows for quiet curiosity without the hype. Knowing that you might be about to delve into a field rife with misinformation, you would naturally want to select articles that would stay true to the science, offering reliable, factual information. With ground news, you can quickly scan the headlines and see which articles are flagged as having low, high, or mixed factuality. This allows you, when doing research on a complex subject, to easily filter out the ones with mixed or low factuality, ensuring you are exposed to the most factual information available on a given news story, allowing for peaceful, informed contemplation. On top of this, when seeking to understand a story, it is valuable to be aware of any potential bias in the reporting. If a story is only being reported by sources that lean to the left, or only by those that lean to the right, that's a pretty significant red flag, something that warrants deeper, quiet investigation before you accept and repeat that information yourself. For this NASA and Mars story, for instance, a tool like ground news might show that 43% of the sources lean left, 29% lean right, and 29% lean center. While this isn't a huge bias, it is there, and this information allows you to pause, to reflect, and to ensure there is no ulterior political motive behind the stories you are reading, leading to a more balanced and complete understanding. Sometimes, the bias is much more pronounced, and ground news helps to illuminate these blind spots with their aptly named blind spot feed. Consider another major science story right now, one about our own planet. The Earth just broke the record for its hottest day, not once, but 2 days in a row. Ground news might reveal that this story is a significant blind spot for the right-leaning media, with only 11% of sources on this critical story leaning right. This feature, browsing the blind spot feed, is particularly insightful as it highlights stories that are disproportionately covered by one side of the political spectrum. It helps you to quietly understand the motives and intentions of various media outlets, making you feel as though you are gently circumventing the wool that so many media outlets might be attempting to pull over everyone's eyes. Ground news, in essence, provides all the quiet tools you need to be a critical thinker, to navigate the often noisy landscape of information with a calm and discerning mind. And at this particular moment in our history, the ability to think critically, to seek out balanced and factual information, has never been more important, never more essential for our collective peace of mind and understanding. It is a service so useful, so vital for navigating the world gently, that perhaps you might consider exploring it further. It's a way to support independent journalism and to foster a more transparent